Welcome back to our next video of our online classes in pharmaceutical microbiology. In the previous classes, we have started this second unit in which we have completed staining techniques. In today's class, we'll be starting about biochemical tests. What do we do in staining techniques? In staining techniques, we impart some color to the microorganism in order to identify them under microscope. It could be either presence of a spore or flagella or nuclear material, or based on if it is gram positive, gram negative, or if it is a mycobacterium species or not. All this can be possible with the help of staining techniques. Other than staining techniques, the other way to identify type of bacteria can be biochemical tests. So what type of biochemical tests are there, and what are their applications, what is the procedure, the principle, we'll be seeing in today's class. So today's class objectives will be to know what IMViC test, okay, what IMViC stands for, that also we'll be seeing in today's class. And after that, we'll be seeing principles in various tests, okay, and after that, procedure and interpreting how we can determine or how we can find which type of bacteria is present. So moving on with IMViC. IMViC, it is a combination of 4 tests. One is I, which is indole production test, M, methyl red test, VI, that is it's a Voges-Proskauer test, and the fourth one, C, is citrate test. So we'll see what all these 4 are. First is I, that is indole production from tryptophan. Tryptophan is an aromatic amino acid. What do I mean by the term aromatic amino acid? It means to the basic amino acid skeleton, an aromatic ring will be attached. Therefore, for this reason, I call tryptophan as aromatic amino acid. Okay, we'll see the structure of tryptophan also. And M stands for methyl red test. What is methyl red? Methyl red is actually a reagent that we use in titrations. What happens is, in a basic medium, methyl red has some sort of color, whereas in acidic medium, it has some sort of a color. In this test, the microorganisms consumes the glucose, okay? Because of consumption of glucose, it forms other compounds or the metabolites which increase the acidity of the solution. As a result, the methyl red indicator changes its color, determining the presence of microorganisms. And next is VI, that is Voges-Proskauer test. It is due to formation of acetyl or acetoin from pyroid. We'll see how it is formed. And last is citrate test. Okay, citrate upon consumption, it gives some other products. Okay, how it is, we'll be seeing in the next coming slides. Beginning with indole production test. Basically, these tests are for Enterobacter species. When I say Enterobacter, always remember these types of microorganisms belong to a particular family called as Enterobacteriaceae. Okay, Enterobacteriaceae family members are tested with this one in order to identify. So Enterobacter species that possess tryptophan, okay, they contain tryptophan within them, can utilize tryptophan. Okay, they contain tryptophanase. They contain the enzyme, and the tryptophan will be present in the nutrient medium. Okay, this tryptophanase, every enzyme has specificity. When I say fructase, okay, this fructase only consumes fructose sugar. If I say sucrase, it will consume only sucrose sugar. If I say hexokinase, it can consume all types of hexokinases, it could be glucose, galactose, and all glucose, galactose, fructose, and all can consume. So there is some specificity for every enzyme, right? Similarly, for tryptophan to be consumed, it needs an enzyme called tryptophanase. If it is a histidine amino acid, for it to be consumed, it needs an enzyme called histidase. Okay. In this case, tryptophan is that is present within the Enterobacter species that will be consuming tryptophan and give out the indication. Okay, so you can see here this is the structure of tryptophan. Okay, this is the basic skeleton of an amino acid, and this one, the bicyclic one, is the indole ring. Okay, what happens is tryptophan is and cause deamination of tryptophan. What do you mean by deamination? Removal of amino group. Okay, deamination means removal of amino group. When amino group is uh, taken or removed from tryptophan, what happens is the indole part gets broken. Okay, suppose this is the indole part and this is the basic skeleton of an amino acid. Okay, when amino group is removed, as a result this one breaks away and this will be the indole ring. Okay, this will be free indole. Presence of this free indole will be giving the indication. Let us see next point. Free indole is detected by para dimethyl amino benzaldehyde. Benzaldehyde, benzene with aldehyde group is benzaldehyde. Okay, when it says para dimethyl amino, it means to the benzene ring. Okay, to the benzene ring which has aldehyde at the top. Bottom it will have amino group. Okay, N group. It is saying dimethyl, so nitrogen will have two methyl groups. So benzene ring, bottom N, two methyl groups, and at the top CHO, that is aldehyde. So it will be para dimethyl amino, para dimethyl amino benz aldehyde. Okay, free indole is detected by para dimethyl amino benzaldehyde, whose aldehyde group reacts with indole forming red color complex. So this para-amino benzaldehyde molecule, the aldehyde part, C double bond OH, this aldehyde part combines with the free indole and forms a complex. That complex gives red color, and then we can determine the presence of that particular type of species of bacteria, that is Enterobacteriaceae. Okay, Bacteriaceae. Next point, let us see. Production of indole from tryptophan is an important biological property of Escherichia coli. Enterobacter, when I say Enterobacter, it means the bacteria which is present in the intestine. Enteron, okay, enterointestine. All the bacteria which survive in the intestine are called Enterobacter species. So E. coli is in our intestine, right? Which helps in digestion. So this test gives positive with E. coli. Production of indole from tryptophan is an important biological property of Escherichia coli. Many strains of group A, B, and C, Shigella, Edwardsiella, Tarda, Klebsiella, Oxytoca, and Proteus vulgaris. All these are the types of, all these are the bacteria which are included in Enterobacteriaceae members. Okay, let us see the procedure of it. First, the bacterial sample is taken. Okay, bacterial sample is taken because it has particular bacteria. We need the bacteria to be tested. That will be added to a nutrient medium. Which type of nutrient medium? The nutrient medium which has tryptophan in that. So if I take nutrient medium having glucose, it will be a glucose broth. Okay. Similarly, if I be specific in terms of chemical substance present, if I want tryptophan, it will be a tryptophan broth. So bacterial sample will be added to the tryptophan broth. Okay, and then incubated at 37 degrees centigrade. The bacterial sample in tryptophan broth. Okay, when the bacterial sample is added to tryptophan broth, so it will be an inoculum that will be kept in incubator at 37 degrees centigrade and leave it for some time. Not some time, it is around 24 to 48 hours. Okay, after adding that, a few drops of Kovac reagent is added. The main component of this method or of this technique is Kovac reagent. That reagent contains para-dimethyl amino benzaldehyde. Okay, bacterial sample in tryptophan. This third one, bacterial sample in tryptophan broth. To this, few drops of Kovac reagent is added. As a result, red ring forms the top. Okay, if red ring is formed, it is positive. As we have seen in the previous slide, When dimethylaminobenzylaldehyde forms complex with indole ring, it gives red color. Okay, that complex will give red color. So the formation of red color will be an indication of the positive reaction. So red ring forms in the top, so it means indole positive. Red ring does not form in the top, it will be indole negative. Let us see how we can interpret. This is the test tube which contains tryptophan broth, okay, and added with a bacterial sample. Upon incubating and adding few drops of Kovac reagent, that is para-dimethyl amino benzaldehyde, it gives red color, okay, and that means it is indole positive, and it also means the presence of Enterobacteriaceae bacteria. If it is negative, that means it will be yellow color. It means there is no presence of Enterobacteriaceae members. Okay. Moving on to the next slide, that is next test, methyl red test. What is methyl red? I said in the introduction slides that methyl red is an indicator used when there is a difference in pH. So let us see what they are. Methyl test is used to determine the ability of bacteria to oxidize glucose and produce stable acid end products. What does it mean? Suppose I take a glucose molecule, and you know glucose molecule has aldehyde at one end, right? Because it is polyhydroxy keto compounds, right? Glucose or carbohydrates are polyhydroxy keto compounds. At the same time, it has COOH at the end of the, or the first carbon. The first carbon of the glucose will have COOH. Okay. When the first carbon will have aldehyde group, polyhydroxy polyhydroxy aldehydes or ketones are the carbohydrates, right, or saccharides. So when it is polyhydroxy aldehydes or ketones, let me consider aldehyde, which is a glucose molecule. Aldehyde means first carbon has aldehyde group. That aldehyde group has C double bond OH. If it undergoes oxidation, it means addition of one oxygen. When COH to it, oxygen is added. What will it be? COOH. So from aldehyde to carboxylic group, it is forming. What is it forming? From aldehyde to carboxylic group. C double bond OH to COOH. It will be a carboxylic group. So from aldehyde, it is forming an acid, right? So there will be a change in pH because of that. We'll see now. Methyl red is a pH indicator as discussed. Red at pH less than 4.4 and yellow at pH greater than 6. If it is more acidic, lesser the pH value, more will be the acidic nature. So up to 7, 1 to 7 is acidic, sorry, 1 to 6.9 is acidic, 7.1 to 14 is the basic. So 7 is the neutral. So in the in the acidic part, lesser the pH, more will be the acidic nature. So if the pH is less than 4.4, then red color will be visible. If the pH is greater than 6, or it could be 6 to 7 or 6 or more than 7, it could be anything. If the pH is less than 4.4, it is red color. If it is greater than 6, it is yellow color. We'll see the procedure as well. For this, the special Newton medium is, what is that? It is a combination of two, and that is methyl red and Proskauer broth. So this methyl red test uses the Newton medium, which is a combination of two, that is methyl red as well as Proskauer broth. Let us see the procedure now. First, the medium is taken, MRVP. Okay. For this, the shortcut uses MRVP, methyl red Proskauer. The shortcut is MRVP. So MRVP medium is taken, okay, that will be kept as a standard. And another MRVP medium is taken. In that we add the microorganism. Adding of microorganism means inoculation of the microorganism. Okay, inoculation. Okay, one will be standard and the other one will be inoculated medium. Now this will be incubated at 37 degrees centigrade for 24 to 48 hours. 24 means 1 day, 48 means 2 days. It could be for 1 day or 2 days. So one we have standard and one we have inoculated. So results, whatever we get, we can compare with the standard. Okay. Now, after incubating for 24 to 48 hours, MR reagent will be added. That is, methyl red reagent will be added. As a result, in the standard, since there was no microorganism in standard, there should be no change in the color, right? There is no change. There should be no change in the color because there is no microorganism to consume that medium. Whereas in the medium in which the microorganisms are present, that will be showing a color. That color, that color indicates the presence of certain type of bacteria. Okay. Uh, you can see here, after adding MR reagent, methyl red reagent, no changes indicates no contamination. No change in color, which it means no contamination, absence of bacteria. If any color change, then contaminant present. If we find any change in color, it means contamination is present. And in this case, there was no color in the standard, and in the inoculated, there will be definitely change in color. So if red color is formed, then it is MR positive. Okay, red color is formed, it is MR positive. If no red color is formed, it is MR negative. You can see the image here. This is the uninoculated. Uninoculated means the standard one, which doesn't have bacteria within it. Only the nutrient material will be present in that. Okay, and this one, let it be some other microorganism is present, whereas this one has the specific kind of bacteria which reacts to MR test. Upon incubating and adding MR reagent, this one is as it is because it has no bacteria, and this one doesn't show any color. It is same as that of the nutrient medium. So this one is showing negative test, and this one is positive. It means the glucose molecule present, which has a aldehyde, that will be oxidized to form acid. Okay, this formation of acid is resulting in decreasing the pH below 4.4. As a result, we get red color. Okay, methyl red basic property is to form red color at lower pH. So this was our methyl red test, and moving on to the Works-Prescott test. What Works-Prescott test? It is basically how it forms non-acidic or neutral material. Let us see the whole description of it, the principle, then we'll understand what it actually is. Basically, this test is used to determine the ability of microbes to produce non-acidic or neutral end products. The previous test, we were able to see red color only if acidic uh, components are formed, that is, aldehyde getting converted to acid. When acidic compounds are formed, we were able to find that particular microorganism, which was showing positive for MR test. Whereas in this case, the components which are formed, which are non-acidic or neutral, will show the different color. Okay. Remember the MRVP broth is a combined medium. Okay. Even in this, the same nutrient material as that of MR test will be used. Okay. The presence of a deep rose color after 15 minutes is indicative of non-acidic neutral metabolic end products and a positive VP test result. Let us see the procedure, then you will understand the last point. First, take MRVP broth. Okay, test tube 1, test tube 2. In the test tube 1, inoculate with test organism, and test tube 2, let it be a standard, that is uninoculated. Incubate for 24 hours at 35 degrees centigrade. Then let us see this one now. After bringing out the inoculated broth from the incubator, take some clean test tubes and to it add 1 ml of broth. Then to it add 0.6 ml of 5% alpha naphthol, then 0.2 ml of 40% potassium hydroxide. Then shake the test tube well and then allow the test tube to remain stagnant for about 10 to 15 minutes. What does it mean? It means after incubating, take a fresh test tube, add a broth into that. Okay. Okay. First, take a test tube, add a broth into that. Then add what are the reagents? Take 1 ml of the broth into the test tube. Then add 0.6 ml of 5% alpha naphthol. Then 0.2 ml of 40% potassium hydroxide, and then shake the test tube well and allow the test tube to stand for 10 to 15 minutes. Remain stagnant means it means let it stand for few minutes. Okay, and after 15 minutes, red color appears. It indicates the presence of diacetyl. Okay, and if no color presents, then negative test. What does it mean? Diacetyl. Okay, diacetyl are neutral or non-acidic, whereas acid are the acidic, isn't it? They cause change in pH. The neutral or diacetyl group are different things. They are neutral, right? Neutral or non-acidic. You can see the last point. The presence of deep rose color after treatment is indicative of non-acidic or neutral metabolic end product and a positive AP test result. You can see here in this image, negative was Posca and here positive. Why is it positive? Because it has red color. And this one is uninoculated, that is the standard. And this one, some other organisms which gives negative test. And this contains the bacteria and shows positive test. Okay, this was Posca's test. The methyl red test was to identify bacteria which is oxidizing glucose molecule and resulting in formation of carboxylic group on the glucose molecule. So it is basically the molecule which are the bacteria which is causing oxidation of some material in order to form acid, that will be found in methyl red test. Whereas in Rose-Posca test, the organisms that are able to form non-acidic or neutral molecules will be found in Rose-Posca test. Moving on to the last one, that is citrate test. When I say citrate, remember Krebs cycle. The enzyme used to form citric acid is citrase. Okay, that citrase will be used in this. Let us see. It is used to determine if an organism is capable of fermenting citrate and using the citrate as its sole carbon source. It means fermenting citrate. It means consuming citrate, and that citrate will only be the sole source for source of carbon for the microorganisms. The ability of an organism to utilize citrate occurs via enzyme citrase. If the organism has citrase enzyme producing capability, if they have the ability to produce citrase, only then they can consume citrate. So the medium for it needs is specifically that which contains more number of citric acid in that. Okay. Citrate is utilized by several of Enterobacteriaceae as a single carbon source. So it is a general point that all the Enterobacteriaceae members consume citric acid, okay, in presence of enzyme citrase. To test this ability, bacteria are incubated in medium that contains only citrate as a source of carbon. Ammonium phosphate is available as nitrogen source. Let us see the procedure now. MRAP medium is taken. MRAP medium also contains citrate in that, okay, and after that the bacteria are added and incubated at 37 degrees centigrade. After incubating, during incubation period, what happens is the bacteria consume citrate, okay, citric acid. When they consume citric acid will be converted to pyruvate. Let us see the reaction here. Okay, this is citric acid. This citric acid in presence of enzyme citrase that is released by the microorganisms forms oxaloacetic acid. You can remember all these. All this comes in the Krebs cycle, right? Oxaloacetic acid, citric acid, fumaric acid, malic acid. All this comes in Krebs cycle. The citric acid gets converted to oxaloacetic acid and acetic acid. This oxaloacetic acid undergoes decarboxylation, gives pyruvic acid. If this pyruvic acid is in alkaline solution, gives acetic acid and formic acid. And if this pyruvic acid, if it is in acidic condition, then it forms acetic acid and lactic acid. So at the end, citric acid is being consumed. Okay, citric acid presence will have one particular color, whereas citric acid absence will have some other color, and that color will be because of the formation of new metabolites. Okay, let us see here. The alkaline byproducts of citrate will cause a pH indicator to turn royal blue. Okay, organisms capable of using citrate as a carbon source turn the media Persian blue. What is Persian blue? It means it is a bluish green color. Okay, that is called Persian blue. So how do we interpret it? These are the two tubes. Okay, one contains the inoculated bacteria and other does not contain inoculated bacteria, or does not contain enterobacterial species but might contain other species. Okay, broth is basically green in color. Okay, this green in color due to change in pH gets converted to bluish green color, that is Persian blue. That interprets that presence of the particular bacteria, that is enterobacteriaceae. Let me repeat it again. In this Vox-Prosca test, MRVP medium is taken into that, the bacteria is added and incubated at 37 degrees centigrade. The MRVP media also contains citrate nutrient material. Okay, this citrate, after adding citrate nutrient material, it will be a greenish color. Okay, this greenish color, in presence of citrase enzyme from the bacteria, consumes all the citric acid and forms its metabolites. As a result, the greenish color gets converted to Persian blue due to formation of new metabolites. This results in color formation of two different colors. If it is completely green, it means absence of Enterobacteriaceae members. Okay, and if positive, it will be a Persian blue, which means you can see the color here. So this is an image of a petri dish containing, left side will be standard and the right side will be inoculated, uninoculated, and the right one will be inoculated. You can see the left is completely green, whereas here it is partially blue or Persian blue. Okay, it is bluish green. Okay, this indicates the presence of Enterobacteriaceae members. So this is it with biochemical tests, that is IMViC test. Let us have a quick recap. First was indole production test. In that, the indole, free indole, when it forms, when it combines with coax reagent, forms red color complex, and the coax reagent is para-dimethyl amino benzaldehyde. Gives red color. The formation of red color indicates presence of bacteria which consumes which produces free indole. Okay, and that means that indole is coming from tryptophan. How is that tryptophan coming? That tryptophan gets hydrolyzed by tryptophanase coming from the bacteria, resulting formation of free indole. That indole binds with the reagent, coax reagent, and forms a complex, and that complex gives the red color. That will be a positive. And next one was IM. M stands for methyl red. Due to change in pH, that is from aldehyde to acid, there will be a change in pH resulting in formation of green color if the pH is less than 4.4 and yellow color if it is greater than 6. If it is less than 4, it means presence of that particular species of bacteria, that is Enterobacteriaceae. Okay, and next was Vox-Prosca test. This was Vox-Prosca test. What does it do? It identifies which type of bacteria. It identifies the type of bacteria which produces non-acidic or neutral molecules. Okay, in presence of MR reagent only due to change in pH. Okay, is it pH? Yeah, after adding the several reagents, that is 5% alpha naphthol, 40% potassium hydroxide, after adding all this, then and keeping it for 10 to 15 minutes, we'll be able to see change in color. Okay, that change in color, if it is red, it means presence of that particular kind of bacteria, the Enterobacteriaceae. If it is not red, it means absence of those kind of bacteria. The last one was citrate test, in which the nutrient medium contains large amount of citrate, due to which the nutrient medium looks greenish color. And after incubating it, if we see only green color, it means negative for citrate test, and if it is positive, it will be a Persian blue. Why is it Persian blue? Because of formation of new metabolites after consumption of citrate by citrase enzyme released by bacteria. So this was it with today's class of biochemical tests of microorganisms. In the next video, we'll be moving with the sterilization topic. Okay, thank you.